Hi everyone, this is Dr. Jeeva. In this video, we will be dealing about the digestion and absorption of the gastrointestinal tract. The major foods on which the body lives can be classified as carbohydrates, fats and proteins. They generally cannot be absorbed in their natural forms to the gastrointestinal mucosa. Therefore, today we will be dealing with the process by which carbohydrate, fats and proteins are digested into small compounds for absorption. First, we will see about the digestion of carbohydrate. Only three major sources of carbohydrate exist in the normal human being. They are starch, which are large polysaccharides present in almost all non-animal food, particularly in potatoes and different types of grains. Sucrose, which is a disaccharide known popularly as cane sugar, and lactose, which is a disaccharide found in milk. Other carbohydrates ingested are amylose, glycogen, alcohol, lactic acid, pyruvic acid, pectins, dextrins, and minor quantities of carbohydrate derivatives in meats. The digestion of carbohydrate starts from the mouth. When food is chewed, it is mixed with saliva, which contains the digestive enzyme ptyalin and alpha amylase, secreted mainly by the parotid glands. This enzyme hydrolyzes starch into disaccharide maltose and some small polymers of glucose that contain 3 to 9 glucose molecules. However, the food remains in the mouth only a short time, so probably not more than 5% of all the starches will have become hydrolyzed by the time the food is swallowed. However, starch digestion sometimes continues in the body and fundus of the stomach for as long as 1 hour before the food becomes mixed with the stomach secretions. Then the activity of salivary amylase is blocked by acid of the gastric secretion because the amylase is essentially non-active as an enzyme once the pH of the medium falls below about 4. Nearly about 30 to 40% starches will have been hydrolyzed mainly to form maltose. After leaving the stomach, the carbohydrate is digested with the help of the enzyme pancreatic amylase. Pancreatic secretion, like saliva, contains a large quantity of alpha amylase that is almost identical in its function with the alpha amylase of saliva but is several times as powerful. Therefore, within 15 to 30 minutes after the chyme empties from the stomach into the duodenum and mixes with the pancreatic juice, virtually all the carbohydrates will have become digested. The carbohydrates are almost totally converted into maltose and or other small glucose polymers before passing behind the duodenum or upper jejunum. The carbohydrate which is converted into maltose, the disaccharide, and small glucose polymers are hydrolyzed into monosaccharides by the help of the intestinal epithelial enzymes. The enterocytes lining the villi of the small intestine contain 4 enzymes. They are lactase, sucrase, maltase, and alpha dextrinase, which are capable of splitting the disaccharides, lactose, sucrose, and maltose, plus other small glucose polymers into their constituent monosaccharides. These enzymes are located in the enterocytes covering the intestinal microvilli brush border. So the disaccharides are digested as they come in contact with these enterocytes. The lactose splits into a molecule of galactose and a molecule of glucose. Sucrose splits into a molecule of fructose and molecule of glucose. Maltose and other small glucose polymers also split into multiple molecules of glucose. Thus, the final product of carbohydrate digestion are all monosaccharides. In an ordinary diet which contains far more starches than all other carbohydrates combined, glucose represents more than 50% of the final product of carbohydrate digestion, and galactose and fructose each represent more than 10%. In the mouth, the juice is saliva and the enzyme responsible is salivary amylase, and the starch is converted into maltose, that is the disaccharide. In the stomach, the gastric juice, the gastric amylase, it is a weak amylase and the action is negligible. And in the small intestine, we have four enzymes: sucrase, maltase, lactase, and dextrinase. And finally, the end product are glucose. So, therefore, final product of carbohydrate is glucose, which constitute about 80%, and fructose and galactose, which constitute about 10%. That's all for digestion of carbohydrates. Now moving on to the digestion of proteins. The important items of diet containing proteins are wheat. Wheat has glutenin and gliadin, which constitute the gluten, and in milk, casein, lactoalbumin, albumin, and myosin; egg, albumin and vitelin; meat, collagen, albumin, and myosin. The dietary proteins are chemically long chains of amino acids bound together by peptide linkage. The characteristics of each protein are determined by the types of amino acids in the protein molecule and by the sequential arrangement of these amino acids. The digestion of protein does not occur in mouth since saliva does not contain any proteolytic enzyme. So the digestion of protein starts only in the stomach. Pepsin is the only proteolytic enzyme present in the gastric juices. Pepsin only initiates the process of protein digestion, usually providing only 10 to 20% of the total protein digestion to convert the protein to proteoses, peptones, and few polypeptides. Pepsin, which is an peptic enzyme of the stomach, is most active at a pH of 2 to 3 and is inactive at a pH above about 5. Consequently, for this enzyme to cause digestion of protein, the stomach juices must be acidic. One of the important features of pepsin digestion is its ability to digest the protein collagen, an albinoid type of protein that is affected little by other digestive enzymes. Collagen is a major constituent of the intercellular connective tissue of meats. Therefore, for the digestive enzymes of the digestive tract to penetrate meats and digest the other meat protein, it is necessary that the collagen fibers is digested. Most proteins are digested in the upper small intestine, in the duodenum and jejunum under the influence of proteolytic enzymes from the pancreatic secretion. Immediately on entering the small intestine from the stomach, the partial breakdown products of the protein foods are attacked by four major proteolytic pancreatic enzymes. They are trypsin, chymotrypsin, carboxypolypeptidase, and proelastase. Trypsin and chymotrypsin are called endopeptidase as these two enzymes break the interior bonds of the protein molecule. Both trypsin and chymotrypsin split protein molecule into small polypeptides. Carboxypolypeptides then cleaves individual amino acid from the carboxyl end of the polypeptides. Only small percentage of the proteins are digested all the way to the constant amino acids by the pancreatic juices. Most remain as dipeptides and tripeptides. The last digestive stage of proteins in the intestinal lumen is achieved by the enterocytes that line the villi of the small intestine, mainly in the duodenum and jejunum. These cells have a brush border that consists of hundreds of microvilli projecting from the surface of each cell. In the membrane of each of these microvilli are multiple peptidases that protrude through the membranes to the exterior where they come in contact with the intestinal fluids. Two types of peptidase enzymes are especially important: aminopolypeptidase and several dipeptidase. They succeed in splitting the remaining larger polypeptides into tripeptides and dipeptides and a few amino acids. Both the amino acid plus the dipeptidase and tripeptidates are transported from the exterior to the interior of the enterocyte. Finally, inside the cytosol of the enterocyte are multiple peptidases specific for the remaining types of linkages between amino acids. Within minutes, virtually all the last dipeptides and tripeptides are digested to the final stage to form single amino acid. These then pass on through to the other side of the enterocyte. More than 99% of the final protein digestive products that are absorbed are individual amino acids, with only rare absorption of peptides and very rare absorption of all protein molecules. This tabular column summarizes the digestion of proteins. In the mouth, there is no proteolytic enzyme and there is no digestion taking part in the mouth. And in the stomach, you have the pepsin, which is the only proteolytic enzyme. And in the small intestine, you have three pancreatic juices, that is trypsin, chymotrypsin, and carboxypeptidase. And in succus entericus, you have dipeptidase, tripeptidase, and aminopeptidase. The final end product of protein digestion is amino acid. Now moving on to the digestion of fat. By far the most abundant fats of diet are the neutral fats, also known as triglycerides. Neutral fat is a major constant in food of animal origin, but much less so in food of plant origin. In the usual diet, or also some small quantities of phospholipids, cholesterol, and cholesterol esters. The phospholipids and cholesterol ester contain fatty acids and therefore can be considered as fats. Cholesterol, however, is a sterol compound that contains no fatty acid, but it does exhibit some of the physical and chemical characteristics of fat, plus it is derived from fats and is metabolized similarly to fats. Therefore, cholesterol is considered from a dietary point of view a fat. Lipids are digested by the lipolytic enzyme. Saliva contains lingual lipase. This enzyme is secreted by lingual glands of mouth and swallowed along with the saliva. So the lipid digestion does not commence in the mouth. This amount of digestion is less than 10% and generally unimportant. Instead, essentially all fat digestion occurs in the small intestine as. The first step in fat digestion is emulsification by bile acids. The first step in fat digestion is physically to break the fat globules into small sizes so that the water-soluble digestive enzymes can act on the globule surfaces. This process is called emulsification of fat, and it begins by agitation in the stomach to mix the fat with the products of the stomach digestion. So fat with the help of bile and agitation in the stomach causes emulsified fat. And this emulsified fat acts upon the pancreatic lipase and is converted into fatty acids and monoglycerides. So the pancreatic lipase is the most important enzyme for the digestion of fat. The other lipolytic enzymes of pancreatic juice are cholesterol ester hydrolase and phospholipase A and B. The cholesterol ester hydrolases the cholesterol ester, and phospholipase hydrolases the phospholipid. This tabular column summarizes the digestion of fats. In the mouth, with the help of lingual lipase, all the triglycerides are converted into fatty acids and 1,2 diacylglycerol. In the stomach, the gastric lipase is the lipolytic enzyme which converts the triglycerides into fatty acids and glycerol. In the small intestine, we have the pancreatic juice, the pancreatic lipase, cholesterol ester hydrolase, phospholipase A and phospholipase B. And in the intestine, succus entericus, you have the intestinal lipids. The final end product of the fat digestion are fatty acid, cholesterol, and monoglycerides. Moving on to the absorption part. Before going to the absorption part, we need to know the anatomical basis of absorption. The total quantity of fluid that must be absorbed each day by the intestine is equal to the ingested fluid plus that secreted in the various gastrointestinal secretion. This comes to a total of around 8 to 9 liters. But about 1.5 liters of this is absorbed in the small intestine, leaving only 1.5 liters to pass through the ileocecal valve into the colon each day. The stomach is a poor absorptive area of the gastrointestinal tract because it lacks the typical villus type of absorptive membrane. This absorptive surface of the small intestinal mucosa showing many folds called valvae convents, which increase the surface area of the absorptive mucosa about threefold. These folds extend circularly most of the way around the intestine and are especially well developed in the duodenum and jejunum, where they often protrude up to 8 millimeters into the lumen. Also located on the epithelial surface of the small intestine all the way down to the ileocecal valve are millions of small villi. These villi increase the total absorptive area around 10-fold. Thus, the combination of folds of Kerckring, the villi and the microvilli increases the total absorptive area of the mucosa to 1000. Extending from the epithelial cell body into each microvillus of the brush border are multiple actin filaments that contract rhythmically to cause continual movement of the microvilli. keeping them constantly exposed to the new quantities of intestinal fluid. Now we'll see about absorption in small intestine and in large intestine separately. Absorption from the small intestine each day consists of several hundred grams of carbohydrates, 100 or more grams of fats, 50 to 100 grams of amino acids, 50 to 100 grams of ions, and 7 to 8 liters of water. The absorptive capacity of the small intestine is far greater than so as much as several kilograms of carbohydrates per day, 500 grams of fat per day, 500 to 700 grams of proteins per day, and 20 or more liters of water per day. The large intestine can absorb still additional water and ions, although very few nutrients. Absorption of water in the small intestine is by osmosis. Water is transported through the intestinal membrane entirely by diffusion. This diffusion obeys the usual law of osmosis. Therefore, when the chyme is dilute enough, water is absorbed through the intestinal mucosa into the blood of the villi almost entirely by osmosis. Conversely, water can also be transported in the opposite direction from the plasma to the chyme. This occurs especially when hyperosmotic solutions are discharged from the stomach into the duodenum. Now moving on to the absorption of ions. About 20 to 30 grams of sodium ions are secreted in the intestinal secretions each day. In addition, the average person eats 5 to 8 grams of sodium each day. Therefore, to prevent net loss of sodium into the feces, the intestines must absorb 25 to 35 grams of sodium each day, which is equal to about 1/7 of all the sodium present in the body. So, whenever significant amounts of intestinal secretions are lost to the exterior, as in extreme diarrhea, the sodium reserves of the body can sometimes be depleted to lethal levels within normal hours. So, the motive power for sodium absorption is provided by the active transport of the sodium from inside the epithelial cells through the basal and lateral wall of these cells into paracellular spaces. This active transport obeys the usual law of active transport. It requires energy, and the energy process is catalyzed by the ATP. The active transport of sodium through the basolateral membrane of the cell reduces the sodium concentration inside the cell to a low value. Because the sodium concentration in the chyme is normally about 142 milliequivalents per liter, sodium moves down this steep electrochemical gradient from the chyme through the brush border of the epithelial cell into the epithelial cell cytoplasm. Sodium is also co-transported through the brush border membrane by several specific carrier proteins, including sodium glucose co-transporter, sodium amino acid co-transporter, and sodium hydrogen exchanger. These transporters function similarly as in renal tubules. Like sodium ions, the other ions like bicarbonate and chloride are also absorbed in the same manner with the help of the co-transporters. The bicarbonate is absorbed in an indirect way. When sodium ions are absorbed, moderate amounts of hydrogen ions are secreted into the lumen of the gut in exchange for some of the sodium. These hydrogen ions in turn combine with the bicarbonate ions to form the carbonic acid, which then dissociates to form water and carbon dioxide. The water remains as part of the chyme in the intestine, but the carbon dioxide is readily absorbed into the blood and subsequently expired through the lungs. Thus, this is also called active absorption of bicarbonate ions. Calcium ions are absorbed into the blood, especially from the duodenum. Iron is absorbed in the small intestine. Potassium, magnesium, and phosphate are also absorbed in the small intestine. Carbohydrate, proteins, and fats are also absorbed in the small intestine. The carbohydrates are absorbed as monosaccharides. Most abundant of the absorbed monosaccharides are glucose, usually accounting for more than 80% of carbohydrates are absorbed. Monosaccharides are absorbed by active transport process. The glucose is transported from the lumen of the small intestine into the epithelial cells in the mucous membrane of small intestine by means of sodium co-transport. And the proteins are absorbed in the form of amino acid in the small intestine by means of sodium co-transport. The digestive end products of the fats are monoglycerides and free fatty acids. These monoglycerides and free fatty acids first become dissolved in the central lipid portions of the bile missiles. Because the molecular dimension of these missiles are only 3 to 6 nanometers in diameter, and because of their highly charged exterior, they are soluble in chyme. In this form, the monoglycerides and free fatty acids are carried to the surface of the microvilli of the intestinal cell brush border and then penetrate into the recesses among the moving agitating microvilli. Here, both the monoglycerides and fatty acids diffuse immediately out of the micelles and into the interior of the epithelial cells, which is possible because the lipids are also soluble in the epithelial cell membrane. This leaves the bile micelles still in the chyme where they function again and again to help absorb still more monoglycerides and fatty acids. The presence of bile micelle causes 97% fat is absorbed. In the absence of bile micelles, only about 40% are absorbed. After entering the epithelial cells, the fatty acids and monoglycerides are taken up by the cell's smooth endoplasmic reticulum. Here, they are mainly used to form new triglyceride that are subsequently released in the form of chylomicrons through the base of the epithelial cell to flow upward through the thoracic lymph duct and empty into the circulating blood. Now, moving on to the absorption in large intestine. About 1500 milliliters of chyme normally pass through the ileocecal valve into the large intestine each day. Most of the water and electrolytes in this chyme are absorbed in the colon, usually leaving less than 100 milliliters of fluid to be excreted in the feces. Also, essentially all the ions are absorbed, leaving only 1 to 5 milliequivalents each of sodium and chloride ions to be lost in the feces. Most of the absorption in the large intestine occurs in the proximal one-half of the colon, so it is named as absorbing colon, whereas the distal half of the colon is essential for storage of feces and it is therefore called the storage colon. The mucosa of the large intestine, like that of the small intestine, has a high capability for active absorption of sodium, and electrical potential gradient created by the absorption of sodium causes chloride absorption as well. The tight junction between the epithelial cells of the large intestinal epithelium are much tighter than those of the small intestine. This prevents significant amount of back diffusion of ions through these junctions, thus allowing the large intestinal mucosa to absorb sodium ions far more completely. That is against a much higher gradient than can occur in the small intestine. In addition, as occurs in the distal portion of the small intestine, the mucosa of the large intestine secretes bicarbonate ions while it simultaneously absorbs an equal number of chloride ions in an exchange transport process. The absorption of sodium and chloride ions creates an osmotic gradient across the large intestinal mucosa, which in turn causes absorption of water. Maximum absorption capacity of the large intestine is about 5 to 8 liters of fluid and electrolytes each day. When the total quantity entering the large intestine through the ileocecal valve or by way of large intestine secretion exceeds this amount, the excess appears in the feces as diarrhea. Apart from ions, large intestine also helps in the absorption of organic substances like glucose, alcohol, and drugs like anesthetic agents, sedatives, and steroids. And that's all for today. Thank you.